Good day, I am Omuka Samuel. This one I'll be doing a video on biochemical test, on microbial biochemical test. Now we know that for every infectious disease that man or human beings or individuals suffer from, there are usually microorganisms that are responsible for that. How are we able to find a specific or individual microorganisms that are responsible for that? That's why there's a need for us to do a biochemical test. Now, before we continue with this, we know that for every sample that we get from an individual that is suffering from a particular infection, that sample can either be subjected to microscopy or microscopic examination, whereby we examine the different staining techniques that are used to identify a particular bacteria. Now, some of the staining techniques that we can actually use to do that, which are very popular to us, we know of the worm staining, we know of the acid fast staining, we know of the negative staining, we know of the spore staining, and even sometimes we do what the motility test. All those things are actually necessary to actually identify the organism based on the stain that they pick up. Now, another thing that we can actually do as well is what we can as well culture the organisms. We can culture them on a specific what culture media, whereby we have the differential media, we have the selective media, even we have the enriched media, even what the enrichment what. All these media are actually available so that we can actually grow some of these organisms in the lab. Now the third one that we'll be doing, which is the biochemical test. Biochemical test. Now, in the biochemical test, we will go into details in this video. Now the fourth thing that you can actually do as well is what? We can look at the serology test, or you can run serology test on that. Now, what do we do about when we talk about serology? Now, we can actually run a glass, let's say, slide agglutination test or tube agglutination test, or as well, we can even do a microscopic agglutination test for that. Now, apart from that, we can as well we can even go even deeper to do what the molecular, look at what the molecular basis of the bacteria, and even do what we call the phage typing. In the phage type, what we try to do is what we are trying to identify the bacteria based on the bacterial phage that are available. So basically, these are some of the techniques that you can actually use to actually identify a particular bacteria in the lab. Now, you will notice that when we just want to focus on these first two, that is the microscopy and the culture in the lab, you notice that we might not be able to get the full knowledge of what bacteria is responsible for a particular infectious disease. For example, in microscopy, let's say for example, an organism that has been cultured in a Mancocchic agar, given a paint color, subjected to microscopy, and it gave us what? A gram negative, bacilli can either be an E. coli, can be a Klebsiella, or can be what? The citrobacter. Now, how can we tell for a truth that gram negative bacilli is what is E. coli? How can you say, or how can we tell that a gram negative bacteria that we have gotten is this particular thing? That is why we actually need what? A biochemical test. Now, what biochemical test does is that it tries to what? To gives an identity to a particular microorganism, even as what? The genus and the what? And the species level. Even getting to what? The subspecies what? Level. So what we are trying to look at in biochemical test, or why we need biochemical test what? We want to identify the organism even to what? To the genus and what? The subspecies, the genus and the species, even getting to what? The subspecies level. That is why there is a need for what? A biochemical test. Now, let's go straight to what we're doing today in the biochemical test. Now, we know that in biochemical tests, as I said earlier, that we are trying to look at the organism, we are trying to classify the organism or identify the organism to what? To the genus and the species level. Now, there are several techniques that we can actually use to identify such organism, which we'll be looking at in this video. Now, I will list them out and then we will take it one after the other. We have the first one, we have the catalase test. Number two, we have the Urease test. Number three, we have the Indole. We have the Indole Production or the Indole Ring test. Number four, we can actually have the Methyl Red test, which can also be called what? MR test. The MR test. Number five, we have the VP test, which is also known as what? The Voges-Proskauer test. Number six, we have the nitrates, the citrates utilizing test. 7, we have the Urease test. 8, we have the Urease test. 8, we have the which test again? You will look at. We have the Urease test. Number 8, we can actually have what again? Yes, we can have hydrogen sulphide test. 9, now you can have the TSI test, which was present triple sugar ion test. 10, we have the gelatin liquefaction test, or the gelatin hydrolysis. Okay, now let's start with the first one, the catalase test. Now, basically, some bacteria has the ability to produce an enzyme called catalase. Now, this catalase acts on hydrogen peroxide, or decomposes the hydrogen peroxide to produce water and oxygen. Now it decomposes this to produce water and oxygen. Now what happens is this: in the catalase test, as this catalase produced by the bacteria decomposes the hydrogen peroxide, it produces what? Oxygen. And this oxygen is seen as what? Pebbles or what? Or effervescence. Now in doing the catalase test, basically what we do is that we take a glass slide, add, let's say, 0.5 mils of hydrogen peroxide to the glass slide, emulsify just a colony or big a colony from your cultured bacteria onto the hydrogen peroxide. If there is what? The production of what? Bubbles or what? Or effervescence, it indicates that these organisms or whatever microorganisms you are seeing has the ability to what? To produce or contain the enzyme catalase, or it has the ability to what? To decompose hydrogen peroxides. Now, the second one that we look at now, an example that we have, mostly example that we have in this what? The Staphylococcus what? Species. They have the ability to decompose hydrogen peroxide to give us water and oxygen. Now let's go to the third biochemical test, which is the Indole Production, or the Indole Ring test. Now, in an Indole Production test, what happens is this: there are certain bacteria that are able to produce tryptophanes. Now, this tryptophanes In the presence of tryptophan, this is an enzyme produced by a particular bacteria. In the presence of tryptophan, convert this tryptophan into what? Indole. That's where the name comes from, Indole Production. They produce tryptophanes, which convert this tryptophan into indole. Now, following or adding a covert reagent to this indole gives us what? Reagent to this indole gives us what? A red complex. A red complex or a red complex or a red coloration. Now, so when a bacteria produces this and is able to act on this to give us an indole, it at the presence of what? A covert reagent, giving us what? A red complex or what? A red coloration. We say that organism what? Contains what? Contains what? Tryptophans. And these are the principle behind what? The indole production or the indole wing test that we do in the lab. So basically, when you want to do this in the lab, what we do is that we pick up a pot in a test tube containing tryptophan. We take a colony of the bacteria into what? The test tube containing that we inoculate and what? Incubate from 24 hours to 48 hours. In the process of incubation or following the incubation, we add what? The covert reagents to it. If there is a color change to what? A red complex or what? To a red complex or a red coloration, that's as a ring on top of the brows, we say that there is indole production, or indole production is positive. But when there is no color change after the addition of what? Of the covert reagent, we say for such test or for the indole production test or the indole wing test, that such particular thing is negative. So basically, that is what we see at what? The indole production test. Now let's go to the fourth one, the Methyl Red Test. Now, in the Methyl Red Test, what usually happens is this: some organisms have the ability to ferment certain sugars. Now, basically in Methyl Red, what you're looking at mostly is what? Is the sugar glucose. Now, there are certain bacteria that has the ability to ferment glucose to give pyruvic acid. Now, initially, this glucose, its pH is neutral. It has a neutral pH. Now, as the organism or the microorganism ferment this glucose, it changes the initial neutral pH to what? To an acidic pH. Now, in addition of what? Of a methyl red, which is an indicator to this new solution that has been produced as a result of what? The fermentation of this glucose by the bacteria, changes what? Gives the new solution in the test tube to become what? Red. As a result of what? The change of what? Its neutral pH to what? An acidic pH. So basically, in the methyl test, what we are trying to look at what? We are trying to look at the ability of what? A bacteria to ferment glucose to give pyruvic acid, changing it from its neutral pH to acidic pH. And when we add what? The indicator methyl red to it, the coloration of what? Of the solution changes to what? Wet, telling us what? That what? There is what? The production of what? Acid as a result of what? The fermentation of what? Of glucose. Now, almost the same thing occurs in what? In the VP test, on the Voges-Proskauer test. Now, what usually happens as similar to what the methyl red is. The same thing happens is this, that in the VP test is this, that certain organisms or certain microorganisms has the ability to what? To ferment glucose as well. But in this time, they ferment it to what? To an acid, of course. Now, following the fermentation of this acid, they metabolize this acid to produce what we call what? The acetyl methyl carbonyl. Now, this acetal carbonyl that have been made by this enzyme, what happens is this: at the presence of what? 40% KOH, that's potassium hydroxide, and 5% alpha naphtol. At the presence of this, at the presence of 40% KOH, potassium hydroxide, and what 5% of alpha naphtol, at the presence of this, they give what? They also give what? A wedge coloration, a wedge complex is formed. As this wedge complex is formed, it indicates that what? That indicates that organism or that microorganism is what? Is VP what? For VP test is what? Is positive for that. So basically, in a VP test, what will happen is that first of all, the microorganism ferments, of course, the glucose with acids. Now this acid is what? Is metabolized to acetal carbonyl, which is this particular acetal carbonyl is what? An acetone compound. An acetone compound. Now, in the presence of 40% KOH, that's potassium hydroxide, and 5% alpha naphtol, this thing gives it produces what? It produces a red complex. A red complex, which indicates that organisms has, or that microorganism has the ability to ferment glucose and is positive for what? The VP test. Now the next thing that we'll look at from what we have done so far, we'll look at what? The citrate test. And even before we go there, now mostly in the Methyl Red and the VP test, we run them most times concurrently. Any of the organisms that usually gives a positive result for a Methyl Red test usually give a negative result for what? A VP test. That simply means that if we have a positive result for VP test, there will be what? A negative result for what? A Methyl Red test. It's run conjugatively. Now, in the citrate test, now what happens in the citrate or utilizing test is this. Now, there are certain organisms that has the ability to utilize citrates. Now, this simply means that in the presence of what? Citrates or sodium citrate, in the presence of sodium citrate, the microorganism utilizes the sodium citrate that is neutral to produce carbonate or bicarbonate. Now, the production of these two changes what? The neutral pH of the citrate, the sodium citrate, which has a neutral pH to what? An alkaline what? pH. Basically. Now, in the lab, to actually run for this, to do such tests, what we usually do is this. Now, we have what? A Siemens agar, or yes, a Siemens citrate agar slanted on a test tube. Now, we saw Siemens citrate agar contains what? Sodium citrate and what? Bromothymol what? Indicator. Bromothymol indicator at its neutral pH is what? Looks like a forest green. Now, when a colony of bacteria has been incubated or have been inoculated and incubated for 24 to 48 hours, when there is a color change from the initial or the neutral pH, the neutral color, which is what? The forest green, to what? A Prussian blue, it indicates that organism has the ability to utilize citrate, to produce carbonate and what? And bicarbonate. Or the presence of bicarbonate and carbonate indicates that the pH of the neutral pH of the sodium nitrate has been converted to what? Alkaline. Hence the change in color of what? Of the indicator. So basically, that is the principle behind the citrate test. Now, when we go to the Urease test, now the Urease test, as the name demands the Urease test. Now, there are certain microorganisms that has the ability to produce the enzyme urease. Now, what it does is that they act on urea, or they hydrolyzes urea. They act on hydrolyzes urea to produce ammonia and carbon dioxide. The hydrolyzed urea, which is neutral, don't forget this. The urea is neutral, has a neutral pH, and the hydrolyzes this. Because of the production of this, they changes from the neutral pH to what? To alkaline. To alkaline pH. Now, because of the change in pH, of course, the indicator that is used in the preparation of such alga will change color. Now, in the lab, whenever we want to test for the presence of urea, we use a slanted agar as well, urea agar. Now, in that urea agar, we have the urea in it, and we also have what? An indicator, or that's what the phenol red indicator, which at its neutral pH, we have its coloration is orange. Yes, it's orange. Now, when an organism has the ability to what? To hydrolyze what? The urea, what happens is this: it changes the neutral pH of being orange to pink, which indicates that organism has actually hydrolyzes the urea to produce what? Ammonia and what? Carbon dioxide, basically. So basically, that is what happens. So when we inoculate, for example, we take a slanted agar, whereby we take a slanted agar and inoculate the colony of microorganisms and incubate for 24 to 48 hours. If there is a production of a pink color, it easily tells us of what? There is a production of what? Of ammonia and CO2, which has changed the neutral pH of the urea to what? Alkaline. So basically, that is what we have in the urea test. For what? Microorganisms. Now let's look at hydrogen sulphide test. Hydrogen sulphide test. Now in hydrogen sulphide test, what happens is this: there are certain microorganisms that has the ability to produce hydrogen sulphide. Hydrogen sulphide, H2S. Hydrogen sulphide. Now what happens is this: this hydrogen sulphide, in the presence of ferrous sulfates and in the presence of ferrous sulfate and ferric ammonium salts, in the presence of these two compounds, including with this, or in the presence of this, they produces what we call what? The ferrous sulfide. The ferrous sulfide, Fe2S. Now, this ferrous sulfide gives what? A black participate, or a black PPT, which indicates that organism has the ability to produce what? The hydrogen sulfide. So when we include, let's say for example, we notice a particular organism, we are thinking we are trying to test whether it can produce what? It can produce hydrogen sulfide. What we do is this: we include this in what? In either a ferrous in an agar containing ferrous sulfate or what? Or ferric what? Ammonium salt. In this, when that organism contains or has this hydrogen sulfide, it converts or it reacts with these compounds to give us what? The ferrous sulfide, which looks or produces what? A black participate or PPT, indicating to us that organism can what? Contains H2S. That is what? Hydrogen sulfide. Now let's look at the TSI. Now, in the TSI, as the name connotes, TSI. Now, TSI simply represents what? Triple sugar test. Sorry, triple sugar ion. Triple sugar ion. Now, that simply tells us that they are triple sugar that is found in this especially this agar, and there is what? An iron in it as well. Now, in this, the triple sugar found in this are what? Glucose, sucrose, and lactose, which are of different concentrations. Mostly this glucose, of course, 0.1%, sucrose 1%, and lactose 1%. Now, why we have this incorporating this? Of course, we know that we are trying to look out for organisms or microorganisms that has the ability to what? To ferment these sugars. That's why basically the reason why they have this incorporated in this. Now, secondly, we have what? The iron. What this iron does mostly is what? Is what? The ferrous sulphate. And if you remember what I just explained, the hydrogen sulphide, the ferrous sulphate helps in the identification of a bacteria that has what? The hydrogen sulphide. So any organisms, or any microorganisms or not microorganisms, any microbes or bacteria that has what? That has what? H2S, that hydrogen sulphide, reacts with what? The ferrous sulphate. With the ferrous sulphate to give what? The ferrous sulphide, which is what? A black PPT that is seen. Now, basically, that's how we do that. Now, in a lab, when we want to run this in a lab, of course, we have a slanted agar, a TSI agar. That contains the triple sugar, one, for checking for sugar fermentation of the bacteria. It contains what? The ferrous sulphate, for checking if the organism has what? The hydrogen sulfide, and as well has this what? Contains what? The phenol red indicator. What does the phenol red indicator does? The phenol red indicator just wants to tell us whether what? There is what? The change of what? pH from either acidic from the neutral pH to acidity, or what? From the neutral pH to alkalinity. That's what this guy does. Now, one of the organisms that usually give us a very good color in this test is the E. coli. Now, when we run a TSI for E. coli, what happens is this. It usually gives us the slant, when we have the test tube. Of course, I told you that the lens is this our test tube. Let me clean some so that we can actually see something here. Yeah. Let's say this is our test tube. Oh, sorry. I'm not trying this one. We have our test tube. Now, if we slanted this, it should be something like this. The agar should be like this. Yes. So that simply means that this will be our slant portion, and this will be our butt. Now, in E. coli, what usually happens is that it usually gives us both the slant and the butt. Usually give us what? A yellow coloration. And sometimes, most times, gives us what? A gas is produced, whereby the agar is pushed or is dislocated or pushed upwards a bit. That's really what happens. That simply means that if it's giving us a yellow coloration, that simply means they have fermented, both the slants and the buds. They have fermented all the three sugars, the glucose, the sucrose, and the lactose. That's why this was a change in what? In color for the indicator of the phenol red. Of the phenol from the original orange coloration to what? To yellow. Now, in the same thing, that simply tells us that since there is no change in what, there was no PPT form, black PPT form, simply means that the organism does not produce H2S. Now, another organism that actually gives us a very, very good color when we want to incorporate some of these things is Salmonella. In Salmonella, what usually happens is this. In the same thing, sorry, let me bring up this. In Salmonella, let's say we have the same test tube and we have this slant. In Salmonella, what usually happens is that we usually have a purple slant. The slant is usually purple. The butt is usually what? The butt is usually yellow. And there is usually what? A black PPT form. Black PPT, precipitate form. No, what that simply tells us is this. Now, that's the production of black PPT means that amino acid contains what? The H2S. the H2 which has acted on what? On ferrous sulfate to give us the black PPT. And the yellow coloration that has been formed at the pot simply means that this organism, the salmonella, has actually mostly what fermented the glucose, since there will be much glucose at the pot as compared as what at the slant. And the what? And the purple formation of the simply means that there is a production of alkaline. Alkalines have been produced. As compared to this case, that has given us a yellowish coloration. Simply means that acid has been produced as a result of the fermentation of the glucose as the pot, giving us what? The yellow coloration, basically. So basically, this is what usually we see, especially in these two organisms. We have the E. coli and what? The salmonella. So we usually try as much as possible to relate this to what? To salmonella. So as black PPT, purple, and although there are some other tests that we can, biochemical tests that we can actually run on it to be sure of what we are seeing. So basically, that is what usually happens with the TSI test. Sorry for my handwriting. Now let's look at the last one before we would call it a day. Let's look at the gelatin liquefaction. The gelatin liquefaction. Now, in gelatin liquefaction, some bacteria has the ability to produce an enzyme called the gelatinase. Now, gelatinase has the ability to act on gelatin to convert it to become what? Very watery, or change it to become what? Liquid form. That's why it's liquefaction. To convert it to what? A liquid state. Now, basically, let's first of all look at this. Gelatin, let's look at the properties. Gelatin acts as temperature beyond 25 degrees. And increasing temperature, that means temperature beyond this 25 degrees, greater than 25 degrees. Gelatin is usually what? Liquid. And temperature below 15 degrees, this gelatin is usually what? Solid. Yes, that's usually what happens. Now, in gelatin liquefaction test, what usually happens is this. When the microorganisms act on gelatin, or let's say that the microorganisms that produce the gelatinase act on gelatin. Don't forget this, that at 25 degrees, temperature above 25 degrees, they are solid, gelatin is solid. And temperature below 15 degrees, gelatin is what? What did I say? Sorry. Yes. At temperature below 15 degrees, gelatin is solid. At temperature above 25 degrees, gelatin is what? Is liquid. So what happens in this test is this. When gelatinase acts on gelatin, what happens is that it breaks down gelatin into what? Into simple amino acids. And in that process, it prevents the gelatin from becoming what? Solid, even at what? 4 degrees Celsius. And you remember that at 15 degrees Celsius, gelatin is supposed to be solid. So even at 4 degrees Celsius, what happens is that when there is gelatinase, as a result of its production by a particular bacteria, it acts on that gelatin, preventing it from what? From being solid. That simply means that the gelatin completely becomes what? Liquid, even at 4 degrees Celsius. So when we see some of this film, when we see some of these characteristics in a bacteria that has been inoculated in gelatin, changing the gelatin that's supposed to be solid at that particular temperature of 4 degrees Celsius to what? Liquid. We say that bacteria contains what? Gelatinase. And this actually gives us what we call the gelatin liquefaction. So basically, these are some of the tests that we run in the lab to identify specific what? Bacteria, to what? The species and what? The genus level. Thank you very much for watching. And I believe that you have learned a lot. I hope that next time that we'll be doing some video, we'll be looking at some of the molecular techniques that we do in the laboratory, some of the DNA sequencing and the PCR methodologies that we actually do in identifying certain bacteria or getting certain characteristics of bacteria. Thank you very much. God bless you.